Today we are going to study mass spectrometry. Mass spectrometry, aka mass spec, is a method of separating and analyzing ions by their mass to charge ratio. Mass spectrometry does not involve a specific region of the electromagnetic spectrum because it is not directly interested in the energies of emitted photons, electronic or vibrational transitions, nuclear spin transitions, etc. So you can be able to see here that these are how ions are separated in gaseous state. So we are only interested in the abundance. What we've just said, that mass to charge ratio. Atomic weights of other elements are defined by comparison. Mass to charge ratio, which is called MZ, the ratio of the mass of an ion to its charge. Molecular ion, an ion consisting of essentially the whole molecule. That's what we call molecular ion. So we can look at the design. We just have ionization source, then we have mass analyzer, and then detector. Very simple. So ionization sources are different and there are very many. If we need gas phase, we have electron ionization, the most very common one. Chemical ionization, we have photo ionization. And then now when we start the likes of electrospray, matrix assisted laser desorption, what we call MALDI, field desorption, plasma desorption, fast atom bombardment, high temperature plasma. So if you've ever heard of ICP-MS, that is when the mass spectrometer is attached to high temperature plasma as the ionization. Electron ionization sources is designed to produce gaseous ions for analysis. So electron ionization, which was one of the earliest sources in wide use for mass spectrometry, usually operates on vapors such as those eluting from gas chromatography. So majorly from heated incandescent tungsten or rhenium filaments. So it can achieve up to 70 electrovolts, and this will turn vaporized molecules into ions. So how electron ionization works: electrons are emitted from a filament made of tungsten, rhenium or etc. They are accelerated by a potential of 70 voltages. The electrons and molecules cross, usually at a right angle, and collide. The ions are primarily singly charged, positive ions that are extracted by a small potential, 5 volts, through a slit. Chemical ionization is a form of gas phase chemistry that is softer, less energetic than electron ionization. In chemical ionization, ionization occurs via proton transfer reaction. A gas for example, methane, isobutane, ammonia, is introduced into the source at 1 torr. Example here is provided for methane reagent gas. You can be able to see here how that comes, and then we get an ion. Time of flight mass analyzers. The principle of time of flight mass analyzers: a batch of ions is introduced into a chamber by a pulse of accelerating current. This chamber has no fields and is a drift tube. Since the ions have the same kinetic energy, their velocities vary inversely with their mass during their drift. Note, typical flight times are 1 to 30 microseconds. Lighter ions arrive at the detector first. The quadrupole can be seen there. It's a very powerful technique. It's one of the simplest and most effective mass spectrometers. So quadrupole analysis, it is easier for an applied AC alternating current field to deflect a light ion than a heavier ion. Conversely, it is easier for an alternating current field to stabilize a light ion. Using this knowledge, a combined alternating current and direct current potential is applied to the rods via the direct current. The ions is attracted to one set of rods and repelled by the other. The direct current serves to stabilize heavy ions in one direction. The alternating current serves to stabilize light ions in the other direction. The ion must pass through the quadrupole to make it to the detector. So the detectors for mass spectrometers, we have the electron multipliers, like a photomultiplier tube. Ions strike a surface, cause electron emission. Each successive impact releases more electrons. Then we have Faraday's cups. Ions striking a cup cause charge to flow across a load. The potential across the load is monitored. So just a revisit on the design. In major cases, we combine gas chromatography to mass spectrometry, or we can also have liquid chromatography combined to mass spectrometry. So when we have gas chromatography combined to mass spectrometry, the gas eluent from a column is piped directly to the mass spectrometry source. When we have liquid chromatography mass spectrometer, The ionization methods themselves serve as interfaces. Techniques like AC, APCI, and AP work on liquid phase samples. The methods are generally tolerant to reverse phase liquid chromatography solvents and some non-polar solvents. Some buffers can quench ionization of analytes though. Bad phosphate leaves are solid upon evaporation. Also ionizes preferentially. Bad, any other non-volatile additives are also bad. But the good thing is that ammonia acetate, formic acid. Another good thing is lower concentrations, less than 50 micrometers. Applications of mass spectrometry. Interpretation of mass spectrometer is the key to the most applications of the technique. Information contained in a mass spectrum, molecular weight via exact or monoisotopic mass, usually obtained through a suitable, accurate measurement of M-ion or molecular ion and odd electron species. Then we have also the protonated or the protonated molecule and even electron species. In some techniques can be confirmed by the dimers, trimers, and other adducts. Molecular formula can also be contained in mass spectrometry, mass spectrum. Ionization energies can also be obtained. Isotopic incorporation, fragmentation, and ion stability. Molecular structural analysis. Let us check at the fragmentation. So fragmentation can also be used to determine structure. Common fragmentation pathways and rearrangements can be predicted in many cases. General rules. Most stable carbocations are most stable fragments. Resonance can stabilize fragments. Loss of small, neutral, stable molecules is favored. A large number of gas phase fragmentation reactions commonly observed in mass spectrometry analysis. Some examples can be able to see there. The rearrangement also occurs. You can also sometimes find rearrangement like the Mark-Lafferty rearrangement is shown below. Molecular, the adducts, typical adducts observed in mass spectrometer. You can be able to see the adducts there, a molecule plus hydrogen, a molecule plus ammonium, a molecule plus sodium, a molecule plus potassium. So for a molecule ion with molecular weight is equal to 400, we would expect the kind of fragmentation as provided there. If you have that molecule adduct with hydrogen, we will find 401. If it is with sodium, we'll add 23. Dimers and other molecular adducts are also observed, as you can be able to see. Molecular structure analysis, isotope patterns. Isotope patterns can be used to determine molecular structure. Example, the well-known method of calculating M+1 and M+2 intensities, especially useful for detecting chlorine, bromine, sulfur, silicon, and many other elements with characteristics profiles. Isotope patterns can also be used to extract out isotope incorporation profiles for labeled compounds. For example, carbon-13, carbon-14, deuterium, tritium, labeled molecules for metabolism studies. Application in isotope chemistry include the detection of stable and radioactive isotopes in synthetic products and in nuclear chemistry. For example, if you look at parachlorobenzophenone, you can look there and see. Molecular accurate mass. Nuclide masses are not integers. Example, 4 things that weigh 28 atomic mass units. So you can be able to see, but they are not integers. We have, so it means that we cannot round them up. So mass to charge ratio measurements to 4 decimal places or higher are needed. Very important. That is very, very important. Accurate mass analysis is often used as a final confirmation of structure or for unraveling complex fragmentations. So you can be able to see atomic mass defects. So more about tandem. Tandem in space means that the mass selection and fragmentation occur in different physical locations with the spectrometer. For example, triple quad, in which we have mass analyzer 1, then there is collision, mass analyzer 2. Tandem in time means that the mass selection and fragmentation occur in the same part of the mass spectrometer but at different times. Example, in ion traps. Application for mass spectrometer experiments, the tandems. A short list of application: mass spectrometers, tandems, studies of drug metabolism, environment samples. Especially useful in drug metabolism because key pieces of drugs can be selected via their products, what we call daughter ions, or their neutral loss characteristics. Mass spectrometer is applicable to any analytical situation where complex overlapping spectra are detected and need to be interpreted. In environmental science, a compound was discovered in smoke derived from burning plant material that increases germination on a range of plant species that typically follow forest fires. And this one you've observed, where even if you burn a charcoal and you grow maize on that same particular ground, you find that the maize there is much greener and bears give good harvest. The compound is 3-methyl 2-fluoro 2,3-C-pyran 2-on, and it was synthesized after being isolated and analyzed by mass spectrometer and NMR. So you can be able to see it here, the 3-methyl, 2-fluoro, 2,3-C pyran, 2-on. You can be able to see it there. Then gas chromatography mass spectrometer was able to detect this butanolide at low levels in smoke waters. The compound is stable at higher temperatures and is active at 1 parts per million to 100 parts per trillion levels. It is derived from a combustion of cellulose. We know that mass spectrometry is also used in proteomics, in study of proteins. So proteome, the group of proteins related to a cell type under certain conditions. Genome, the complete DNA sequence of a set of chromosomes. Proteomics, the analysis of native and post-translationally modified proteins to characterize complex biological systems. There are at least 3 types of proteomics. Very hard to pronounce that word. Profiling proteomics identify the protein in a biological sample. Then functional proteomics determine protein functions by finding specific function groups or interactions. Then structural proteomics determine the tertiary structure of proteins and their complexes. Then we have sequencing, the analysis of primary structure of an oligomer by bottom up. Can use processes like chemical digestion, then mass spectrometry, or top down to do it all via selective fragmentation in the mass spectrometer. Mass spectrometry is used as a routine chromatography detector, especially in gas chromatography and liquid chromatography. So 2 modes: single ion monitoring, observe 1 to 4 ions selectively, improve signal to noise for ions of interest, total ion current, sum of all ions can be noisy but also captures potential unknown mass to charge ratios. In these cases, the basic mass spectrometer system, usually simple quadrupole with limited resolution and mass ranges, is known as the mass spectrometric detector or mass spectrometric detector, MSD.